everyone. My name is Max Vargas, and I am a fourth year neuroscience graduate student here at UC Davis, working in the lab of David Olson, where I research psychedelics, a really promising class of compounds that has been shown to have powerful antidepressant effects in both rodents as well as humans. Recently, our lab published a study that these compounds are able to promote neuroplasticity, or the way that the neurons in our brains are wired together. And what you can see here in this column of 3 neurons, at the bottom is a neuron that was treated with DMT, a classical psychedelic. What you can immediately see is how much this neuron grew when you compare it to the vehicle control. It's quite striking. It grew way more. But what was interesting to us is that when you look at 5-HT or serotonin, when the neuron is treated with this compound, it doesn't display the same amount of growth. It's basically the same as vehicle. And this was really interesting to us because both serotonin and DMT are agonists of the serotonin 2A receptor, which is the principal receptor believed to mediate the ability for these compounds to induce neuroplasticity and potentially the therapeutic effects. And we began to think this might be due to the localization of the receptor. And on the right here, you can see a group of hex cells, and below it, a cortical neuron. And when you get the hex cells to express the 2A receptor, you can see that it's primarily localized along the plasma membrane, where you would expect the GPCR to be. But in cortical neurons, it's quite different. It's expressed intracellularly, primarily by the apical dendrite. And this begins to explain why serotonin can't promote plasticity. It's a membrane impermeable compound. It can't cross the plasma membrane, and it can't gain access to its intracellular pool of receptors. So to test this idea, we did some clever chemistry where we developed membrane impermeable analogs of psychedelics. So in red, we have the membrane permeable versions, DMT and psilocin. And in blue, we have the membrane impermeable versions, TMT and psilocybin. So what we initially did was we treated the cells with all of these drugs and measured how much they grew. And that's displayed here in this bar graph, where the taller the column, indicating the more that neuron grew. And what we can immediately see is the membrane permeable compounds, DMT and psilocin in red, were able to promote plasticity really well. They grew way more than the vehicle control. But when we look at the membrane impermeable compounds, TMT and psilocybin, there wasn't much of a change when you compare to vehicle. But what we really wanted to show was that if you can get these compounds into the cell, the membrane impermeable ones, then they would be able to promote plasticity just as well as a membrane permeable compound. So to this end, we used electroporation, which is a really cool technique that induces an electrical field around the cell and creates these temporary pores through which drugs or other material can enter. And then the cell heals with now having the drugs inside. So when we did this, what we saw was really fascinating. So what we saw was that the membrane impermeable compounds, when electroporated into the cell, were now able to promote plasticity just as well as the membrane permeable versions. So here you can see that TMT and psilocybin in blue promote plasticity about just as well as the membrane permeable versions, DMT and psilocin. And you can compare this side by side, and it's quite striking. You can clearly see that when you electroporate in the membrane impermeable compounds, they can promote plasticity just as well, showing that you need to stimulate this intracellular pool of 2A receptors in order to induce neuroplasticity. And further understanding this phenomenon is really important in aiding in the development of future psychedelic-based analogs that can be used to treat depression as well as other neuropsychiatric conditions. Thank you for your time, and I appreciate you listening.